So it's my great honor to introduce the speakers for the second session of CCF Research Fellowship Reports. Our first speaker of this session is here live today, Dr. Mark Yarchoan, who is an assistant professor of medical oncology at Johns Hopkins in Baltimore, and also runs an NCI-funded laboratory, as well as leading multiple clinical trials of immunotherapy and novel combinations thereof for patients with hepatobiliary cancers. So he'll be speaking to us today on his fellowship project entitled Deep Immune Profiling of Intrahepatic Cholangiocarcinoma. Mark?
All right, thank you. Thanks to everyone who hung around for this. Let's see if I can make this work. So these are my disclosures. I'm going to be talking to you about the immune microenvironment in cholangiocarcinoma, specifically in some molecularly defined subsets. So just a little bit of background. Cholangiocarcinoma is not one cancer. I think everyone here knows this. It's actually many different cancers that all share a sort of anatomical origin. And we know that immunotherapy provides some clinical benefit for patients with cholangiocarcinoma, but perhaps not as much as we would have hoped for. And one of the strategies to improve response rates to immunotherapy is to really understand the immune microenvironment of specific immune subsets of cholangiocarcinoma, which may be able to inform combination strategies in those subsets. So the first thing we did was we partnered with Tempus. Tempus is a company that does DNA and RNA profiling of cancer, including cholangiocarcinoma, as standard of care. And so they sit on this sort of treasure trove of data, a lot of which is not publicly available. And they were very generous and shared data with us. So as you can see here, every single vertical line is a patient with cholangiocarcinoma. We looked at about 400 patients, which is actually the largest cohort of patients with cholangiocarcinoma that's ever been profiled with DNA and RNA, as far as we know. And you can see that we did some cluster analysis and found that there really were 4 molecularly defined clusters of cholangiocarcinoma in our analysis. There have been other cluster analysis of cholangiocarcinoma. Ours, as you can see, has slightly different clusters than some others, perhaps because these are purely US patients, and so not as much fluke-associated cholangiocarcinoma. So cluster 4, all the way on the right, this is the group of patients who have mostly FGFR2 driven cholangiocarcinoma. Cluster 3 is a basket of mutations that we know can affect tumors through chromatin remodeling, epigenetic changes. So IDH1 mutated, ARID1A mutated cholangiocarcinoma, and others. Cluster 4 was purely intrahepatic cholangiocarcinoma. The other clusters were mixed, both intrahepatic and extrahepatic. And then we looked at the immune microenvironment of these clusters, looking at the RNA data that had been obtained from these patients. Again, I only have a 10-minute talk, so I can't possibly go into all this data, but this is a broad overview. What you can see is that cluster 4, which again was sort of the intrahepatic FGFR-driven group, really had the most immune-excluded microenvironment by RNA, the lowest PD-L1 expression. By contrast, this cluster number 1 was sort of the most immune-inflamed subset. And we looked, so this is a summary of the subsets again. Cluster 4 being intrahepatic, FGFR-driven, had the lowest PD-L1 expression. The most inflamed was this cluster 1, with the other 2 clusters being in the middle and having some unique features to each cluster. We then looked beyond the clusters at individual mutations. Many of these I'm sure people will recognize. Again, I have limited time, so I can't possibly go into all these mutations, but I want to call attention to just 2 of them. So IDH1 mutated cholangiocarcinoma is a distinct subset I think that we now recognize as druggable. And interestingly, this subset had lower B-cell infiltration, tended to be less inflamed, but actually had more M2 macrophages, which we think about as being a sort of immunosuppressive immune subset. And then FGFR2-driven disease had the lowest immune infiltration of any of the immune subsets by RNA. So the advantage of gene expression profiling like this is that you can look at endless numbers of markers. But we really think about this as being more exploratory because RNA doesn't always translate into protein, and you don't really know what cell subset in the microenvironment is making the marker that you're looking at. So we really wanted to confirm our data with some sort of proteomic-based assay. And so initially we proposed doing CODEX, which is a multiplexed imaging platform similar to immunohistochemistry, but with many different markers, looking at samples from our cohort at Hopkins. But when we started this project, we were actually connected with Enable Medicine, which is a company that is interested in producing a sort of tumor atlas for cholangiocarcinoma. We were connected to them actually by Stacy and the CCF, and so we partnered with them to look specifically at a couple of molecularly defined subsets of cholangiocarcinoma, including IDH1, FGFR2, and other subsets of cholangio. Excuse me. So this work is ongoing. I just want to give a shout out to Marina, who's here, who's a junior faculty member at Hopkins and is leading this work with me. And she took the time to really collate these patients, look through their clinical histories, and make this biobank that we've been using. Again, this is very preliminary, but on the left is an IDH1 wild-type intrahepatic cholangiocarcinoma, and this is the microenvironment using this CODEX platform. CODEX can stain dozens of different markers, but there's no way for our human eye to make any sense of dozens of different colors, so we've sort of deconvoluted this and only shown a couple of different markers. And then on the right is an IDH1 mutated cholangiocarcinoma. These are pretty representative images. And what you can see on the right is, it's a little bit hard to see, but there's a bit more red dots. Those red dots are staining CD163, which is an M2 macrophage marker. So it's sort of going along with what we saw with the RNA, that there seems to be more staining of these immunosuppressive or M2 macrophages in IDH1 mutated cholangiocarcinoma. And here's another image. Again, hard to see, but there's a bit more red that you can see on the right, these red dots staining this low prevalence but quite immunosuppressive, we think, subset. So this work is ongoing. We're still working to quantitate these images. I hope to have an update for you if I'm invited back, but at least so far the CODEX is not only feasible but seems to be recapitulating some of what we saw with the RNA analysis. And of course, I'm a translational researcher, and so I really believe that mouse models can confirm findings and answer mechanistic questions in ways that these sort of correlate studies cannot. So this is work from Dan Zabransky, who is an oncology fellow in our group, and I think will be a rising star in cholangiocarcinoma, so someone to watch. But he took a cholangiocarcinoma cell line that was courtesy of Greg Goris, who's here, the SB1 cell line, and actually CRISPR'd in IDH1 mutations into the cell line. You can see that consistent with human IDH1 disease, that these tumors make the oncometabolite that you expect, the D2-HG, at quite high levels. And he implanted these tumors into the livers of mice orthotopically, let them grow up, and then resected both the wild-type cell line and then also the mutated cell line to understand how did adding an IDH1 mutation alter the tumor microenvironment. And once again, very consistent with what we saw with the human data, we saw both lower CD8 T cells in the IDH1 mutated subset, and then quite higher M2 macrophages using CyTOF, which is another profiling similar to flow cytometry. So, in summary, it's a lot of information in 10 minutes, I know, but we've profiled many tumors using many different means to really understand the immune microenvironment of molecularly defined subsets of cholangiocarcinoma. And by RNA, using profiling, we find that IDH1 mutated cholangiocarcinoma and FGFR2 mutated cholangiocarcinoma really do have unique microenvironments with more M2 polarization of macrophages in the IDH1 mutated subset and significantly less immune infiltration in FGFR2 mutated disease. Our CODEX analysis is ongoing, but seems to show similar trends thus far. And actually, our mouse data also recapitulates these findings. So this is very preliminary, but I think that these data provide, I think, some confirmation that cholangiocarcinoma does not have one microenvironment, but it has many different microenvironments. Each of these molecular subsets are unique, and it may eventually become necessary to develop unique treatment strategies for immunotherapy in these unique subsets. So thank you very much for the support. Thanks for the opportunity to talk, and happy to take questions. Oh, sorry. Most important slide of all, just quick acknowledgements. I just wanted to, first of all, thank Neela Azad, who's really the reason that I'm working in cholangiocarcinoma. She's been my mentor since I was a fellow. I want to acknowledge Marina, who we were so fortunate to bring on faculty, who's doing a lot of the CODEX work, along with a team from Enable Medicine, and then Dan Zabransky, who's in the lab with me, and someone to watch.